Okay, my name is Iran Ali. I am a chemist. My background basically is chemistry, so I am a chemist by profession. And this research study has been done a couple of years ago, but is still relevant. And basically, it was the investigation of bioactive properties in a selected plant species from the Legumiaceae plant family found in Guyana. Now, this plant family, hasn't no work has been really done on it since the plant family around the world is very wide. And basically, the Legumiaceae plant family is large and economically important family flowering plants, which is commonly known as a legume plant family, which is a pea, bean, pulse, which we normally eat on a daily basis. Now, basically, plants are the first source of medicine that was used for both human and animals, and even now at present, we still use them to see, what medicinal property and what use we could have from them. And to basically derive somewhat basic way of deriving properties we can get is using bioassays to check for these properties. Basically, there were three objectives in this research, and the first is to investigate the bioactive properties in the selected species from the Legumiaceae plant family using cytotoxicity bioassays. And the second is to prepare crude extract from the selected bioassays, and then the third to determine which solvent is most effective in extracting the bioactive properties from the plant species. Basically, bioassays is basically or assaying generally is extracting a certain compound or a certain type of molecule from the plant um, parts to see what the what you're basically looking for, the properties you're looking for. So it's basically the assay is just a simple method of extracting what you need to check. All right. From literature review, extensive work has been done on the plant family worldwide, but none has apparently been done in Guyana. I've checked with the biodiversity of the University of Guyana, and they have really got no information on the plant family that I have selected. Now, these plant families, they are the Fabaceae or the Legumiaceae, whichever you would like to call them. These plant families, they have uses for such as antiviral, anti-Parkinsonism, anti-inflammatory, paralysis, respectively. This is because they contain secondary metabolites, namely alkaloids, aromatic substance, and terpenoids, which are used in medicine today. Right? These are from literature review. These are some of the compounds which we could simply extract from plants, which are ephedrine, and morphine, which is used extensively in the medical field worldwide. Now, the methodology of carrying out this investigation was first the collection and extraction. The collection was done at the local vicinity at a school where there was a large tree, and there were basically four parts that were selected, which was namely pods containing the seeds, the barks, the stems, and the leaves. These were collected and then they were air dried. After air drying completely, then they were placed with the different, which there were 200 grams was air dried, and each was placed in large jars, and then they were submerged with the two different solvents, one dichloromethane and one methanol, and each of these is to extract the bipolar and non-polar constituents of the parts of the plant. Then secondly, the preparation of the brine shrimp larvae. Basically, this was a simple mechanism that was placed. Is you prepare seawater with a special salt bought from the aquarium place, and basically the larvae were placed in the solution, and after 48 hours they would be hatched and they would be ready for usage. Then last but not least, the brine shrimp microbial cytotoxicity assay. Basically, after the extraction of the from the plants, three different concentrations were prepared, which was 1,000 ppm, 100 ppm, and 10 ppm. And then the brine shrimps were placed. 10 was placed in each different concentration, and after 48 hours, the death toll was checked, how much survived and how much did not survive. And these were the results. So basically, from the results, the two different solvents, dichloromethane and methanol, When in the different vials 1, 2, and 3, the different concentrations, they were checked for the amount of survivors of brine shrimp in each vial. So from each part, the first being the bark on the 4-solvent dichloromethane, in the 10 ppm, in the lowest concentration, there were survivors. In the first two concentration, 1000 and 100 ppm, there were no survivors. For the same, for the leaves for the dichloromethane, in the first vial, there were survivors. In the second and third, there was no survivor. And as you can see, the results continue. In the stems, in the first two concentration, there's little bit of survivors. In the pods, at the lower concentration, they're mostly survivors. Continuing into the methanol solvent extraction at a different concentration and different parts of the plant family. As you can see from most of the results, the death toll was, 100%. Now the calculation is as follows. Percentage death is died in tests and number died in control and number exposed. And it's a simple calculation. Now, the SPSS program we used to analyze the data, but since the percentage death was so high, it could not be computed using the program. There was 100% death of the brine shrimp in the controls using the solvents, hence, the solvents are toxic to the organisms. From the table, it can be seen that at concentration level of 1000 and 100 ppm, there was 100% death of the brine shrimp from both solvents. This can be linked to the fact that this plant family contained bioactive properties, secondary metabolites, such as alkanides, aromatic compounds, phenolic compounds, which is according to literature. For the methanol extracts, only one brine shrimp survived at concentration 10 ppm for the pod, stem, and leaves. This may be due to the fact that methanol was used to extract the polar constituents, which are more toxic to the larvae compared to the dichloromethane extract at concentration 10 ppm for all the plant parts. There were more survivors, thus the non-polar constituents are less toxic at concentration below 10 ppm. These are some of the recommendations for future work, which can be carried out is by using solvents such as ethanol and hexane instead, and also lower concentrations of less than 10 ppm, which can be used as on the brine shrimp. And concluding from the bioassay carried out using the brine shrimp lethality, it is conclusive that the